Good evening, dear students. Today we have come here to discuss certain things about control of microorganisms. And in the studio, I myself, Dr. Rakesh Patel from R.G. Shah Science College, and with me I have a colleague, Professor Rikin Parikh from Arts Commerce Science College, Khambat. We are going to discuss about the control of microorganisms with specific reference to the chemical agents. That is, how to control the organisms, its multiplication and its propagation with the means of certain chemicals. This is a very important topic from the viewpoint that we take lots of medicines, we use lots of chemicals, we use lots of substances either in the laboratory or at home in order to get rid of these organisms. Now let me make it very clear to you that for a layman it may seem that whenever a reference to bacteria or a microorganism is made, they are always harmful, but it's not so. Very little percent of the lot is rather harmful, and we call them as pathogenic. So we are interested in controlling these kinds of organisms, and much of our research has been put into the field of controlling the organisms by one way or the other, specifically the pathogens, because they are harmful to us. So we are going to discuss in detail the chemical methods to control the organisms. Here we are going to take into considerations those chemicals which we are using it in the laboratory and at the home, but not for a personal use, that is not as a medicine, that we call them as a drugs or antibiotics. Those are also the chemicals, but we are not going to discuss that aspect at present. We are going to discuss about the routine chemicals, the chemicals which are used for purposes other than the medicines. Okay, before we start the topic, let us understand certain definitions, because these words will be used many times during our discussions. So to begin with, the first one, we are going to study the definitions. What do you mean by sterilization? Sterilization is a treatment or a process which either kills or removes all living cells of all types. When I say all living cells of all types, that includes the smallest one, including viruses, and those which are very resistant, that is the spores. So any living form, any life form of any type is either killed or it is removed. Now the difference I would like to tell, say for example a filtration method, it removes the organisms, while when we autoclave, when we use physical methods like heat, then radiations, these are the physical methods by which organisms are killed. And there are certain methods by which we remove or separate the organisms from the object, like filtration from a solution. So that is what a sterilization means. So when we say a product is sterile, that means that it is safe. No organisms of any kind is present in it. We will see the different meanings of, this is the literal meaning of sterilization. The other meanings of the sterilization we will see at a later stages, because in industries or at certain points these definitions may have certain variations depending upon its aims and objective of the technique. The second one is disinfection. Now disinfection is a treatment which is usually given to any substance or any material whereby the number of organisms will reduce either on the object or on the surface. But that does not mean that it is going to kill the organisms. It is reducing the number of organisms, and thereby the probability of getting an infection or probability of getting sick is less. So this infection, the very common example is say at home જ્યારે આપણે ઘરે ફિનાઇલ નાખીને પોતું લગાવીએ છીએ, તો that example is a kind of a disinfection, or a doctor using a alcohol swab on our finger or on our hands for taking up a blood. So that is a kind of a disinfection whereby the number of organisms are reduced in number. Next is sanitation. Sanitation means we are reducing the number to that extent or to that level whereby the product or the material is considered to be a safe for the consumption. That is known as a sanitation. So we say that the material has been sanitized. So it is a reduction, or we are reducing the number in such a way that the product becomes a safe. Then comes antiseptic. It is a kind of a disinfecting agent which is normally used for our body. So the example which I gave you earlier, that is using a phenyl for cleaning up of our or swabbing or sweeping up of our floor, you cannot call it as an antiseptic because it is used for a floor. However, when we used alcohol swab for cleaning our fingertips or cleaning our veins, surface of our skin, that is an antiseptic. There are lot many antiseptics available in the market. One of them is iodine solution also. Then we have a term which is known as a SIDL, bactericidal or viricidal. Cidal is basically a suffix which is added to the word at the end, which means anything which we are killing. So whenever a organism is killed or a material which kills the organism is known as bactericidal, it means it kills the bacteria. Next is static. It means it is not killing the organism, but it is inhibiting the growth of that particular organism. Say for example, fungistatic agent. It means that a compound or a chemical is able to inhibit the growth of the organism. The basic difference between SIDL and static is that kills the organism, and it means that once the organism is killed, it is not going to recover anymore. While in case of static, the organism is inhibited as far as that particular agent is present in or around that environment. Once that agent is removed, the organism may recover and organism can again start multiplication. So static is basically a temporary phase where the organism stops reproducing or its metabolism is checked. Then next comes sepsis. Sepsis means a contamination. This is a very common word which we use it in the laboratory. It means that a contamination of microorganism. So that is known as a sepsis. Sepsis may lead to problems in the laboratory, contamination in the plates or in the media, laboratory media, or sometimes it may also lead to kind of a skin infections. Then opposite of sepsis is asepsis. It means lack of bacterial contamination. Where there are no organisms in or around the vicinity, we say that it is a aseptic condition. These kinds of conditions can be achieved by different ways. Now, before we go into the discussions of individual chemical agents, let us discuss certain things about the microbial death. Now, what do you mean by when I have told you in the beginning that a definition of sterilization has different meanings at different places. Here we would like to discuss few variations in that definitions. Basically, we are going to kill the organisms. When we are using some chemicals, we see to it that organisms are killed. That is the most effective chemical agent. that leads to the death of the organism. Now, what do you mean by death of the organism? Like in human beings, when we say a human being is dead or an animal is dead, you already know what the death is. But in case of microorganisms, it has a various kinds of meanings. For example, the first one, it involves the permanent loss of reproducing capability, even under the optimum growth conditions. That is, even the best conditions are provided, all favorable environment is present, the organism is not able to reproduce. Though it may be living, but still it is not able to reproduce. That is also considered to be a kind of a death. Second is the organism is alive, but it cannot be detected by certain procedures. That is, it is not able to be detected by the procedures like cultivation, or in third case, it may also involve the loss of movement. The organism becomes still. Now, when we see that these all things, we call it as a death of the organisms. But what are the conditions which influence the antimicrobial activity? Antimicrobial activity, when we say, it means those chemicals which are able to either inhibit or they are able to kill the organisms. What conditions will influence them, or what are the parameters, what are the factors? Then we should always remember that the entire microbial population is not killed immediately. Immediately means just in a one go or in a one moment, the population will not be dead. Instead, the death of the organism is taking place over a period of time. It takes certain time to kill those organisms. When we are talking this, you should always consider that we are not talking about, we are not mentioning, or we are not concentrating on a single organism. We are talking about the population of microorganisms present in that environment or on that material. So, this death of the population over a certain period of time is always exponential. Now, what is that exponential? It is something which is similar to the growth phase which you have studied when you are studying the cultivation and the growth of organism. In this case, the bacteria normally die at a constant rate. If you see this in the graph, then the graph is plotted logarithmically, and this will always give you a very straight line. On the x-axis, it is the time 1, 2, 3, 4 in minutes, and on the y-axis, it is the logarithm of number of microbial survivors. So, as you can see in the graph, this decrease in the population over a period of time is a logarithmic. It is decreasing by 90%. One log decrease, it means 90% of the population is killed. In this way, if you consider, theoretically, one can never achieve 100% sterilized conditions. That is, if you consider it as a logarithmic decrease, then there may be a 0.1 or 0.01 or 0.001% organisms present. Now, when it goes beyond decimal, it means that we consider it in a different way, that one organism, you can understand what is meant by one organism surviving. It means one organism is still there. But when it is a 0.1, it means that in a volume of 10, there will be one organism. When it is 0.001, it means in a volume or in a unit of 100, there is a survival of one organism. In this way, theoretically speaking, sorry, we never achieve a complete sterilization. And this is what the industrial units or in industrial fermentations is a very vital and is considered. So, in industrial fermentations, the media as a such, because there we have to sterilize thousands of liters of media, you do not achieve a complete sterility. Economically also it is not feasible, because that will always lead to a huge consumption of electricity or a power, and product produced will not be competitive as far as the cost factors are concerned. So this is how the rate of microbial death is defined and it is understood. Now, what are the factors which affects the effectiveness of the antimicrobial agent? Now, I would like to explain it in a different way, in a sense that whenever we are talking about antimicrobial agent, in actual conditions, in actual field, the antimicrobial agent and the organism, when they come in contact with each other, the conditions will not be optimum. There will be many factors, many variables which will play an important role. Some of them, they have been listed here. That includes population of the organism. Naturally, greater the load of the organisms, the sterilization or the effectiveness of antimicrobial agent will be decreased. These are both inversely proportional. Then the type of organisms. We will see at a later stages the resistant patterns of different organisms, because all organisms are not sensitive enough. Some of the organisms may take a lot time, and while the others, they are delicate enough and can be killed instantly. Then comes the concentration of antimicrobial agent. To what extent we should use this antimicrobial agent? Should we use it in a concentrated form or should we use it in a dilute form? Because this is after all economic concern. If we are going to use it in a diluted form and still it is effective, then well, that agent is considered to be a cheaper one. And if we are going to use it in a concentrated form, the effect may be better, but still it will not be very cost effective. Third factor is the duration of exposure. How much time it takes for a destruction of the organisms. It may range from few seconds to minutes. Then other physical factors like temperature of the prevailing environment, pH, presence of organic matter, these are other variables, factors which play important role. The last one is biofilm formation. Biofilm is a kind of a slime layer which is formed by a certain group of organisms where the microbial population is in very large number and it is covered by a certain polysaccharide, microbial polysaccharide, which leads to the kind of a sticky film. The ecology of biofilm is entirely a different aspect which we will not be discussing here, but it is believed that organisms present in a biofilms, they are better protected when they are exposed to certain physical or chemical harsh environments. So these are some of the factors which affects the effectiveness of an antimicrobial agent. Now, as I discussed earlier, one of the factor was the sensitiveness of the organism, how far that organism is susceptible. This graph or this chart shows the resistant pattern of the organisms. At the top we have prions. Prions are proteinaceous materials which multiplies and cause certain diseases like mad cow disease or Kuru diseases. They are considered to be very resistant. Next comes the spores, endospores of the bacteria. Then we have in line certain organisms like tubercle bacilli, that is mycobacteria. Then we have cysts of the protozoas, vegetative protozoas. Then comes gram-negative bacteria, then fungal spores, then viruses having certain envelopes. Then we have gram-positive bacteria, and last, for example, the most sensitive one is the viruses having the lipid envelopes. So these are the basically the organisms which I have mentioned in a descending manner, which shows the resistance capabilities of the organisms. So as far as bacteria is concerned, the sporulating bacteria, endospores, are considered to be very resistant and they are difficult to be killed. Now this chart, probably you may not be able to see them, but this chart shows the various microbial control methods. Basically we have divided into three categories. The one is physical agents, which is mentioned in pink color. Then we have chemical control methods, which is there in a blue color, and then mechanical control methods. We are at present going to discuss in detail about the chemical agents, and the further discussions of various chemical agents will be taken up by Professor Rikinbhai Parikh. Yes, Rikinbhai.
Thank you, sir. And good evening to all friends and viewers. As Dr. Rakeshbhai Patel has mentioned the different basic terminologies regarding this chemical agents and the terminologies which are going to be used by us or by me in the further discussion. So I think now you all are been very clear about the basic fundamental details of these terminologies. So keeping that in mind, we will move further that how these particularly chemical agents will be utilized for controlling the growth of microorganisms. The chemicals that we will divide them basically in two parts. First of all, that the chemicals that are going to do sterilization and the chemicals that are inhibiting the deteriorations. So sterilization, as Sir has mentioned earlier also, that is the complete absence of any kind of microorganisms. So the use of disinfectants, antiseptics and sterilants, they will going to do this sterilization, and the organisms the agents which are going to do the or inhibit the deterioration, that is the demergers and all. So next we will look that the level of chemical decontamination, that what should be the level of chemical agents should be used for the process, that is the high level germicides. If we are going to use high level germicides, then they are going to kill endospores. That is very important, that when we are having such type of the environment or such type of the material in which the endospore might be present, then it is usually not that much easy to kill them or to kill the endospores in comparison to the vegetative structures of bacteria or any other type of organisms. In that type of the conditions, we have to use high level germicides. The devices that are not heat sterilizable and intend to be used to sterile the environment means what? That the devices or the materials which are not sterilized by some physical agent or physical aspects, in that type of the condition we have to use this type of the high level of germicides. So this high level of germicides should be used in that type of the conditions. Now the secondly, that is the intermediate level, that if that much amount or level is not to be used, then intermediate level should be utilized. In this type of the conditions, they will kill the fungal spores, not endospores. There is a basic difference between fungal spores and endospores. The difference is that fungal spores are utilized for asexual mode of reproduction and endospores are the resisting structures. This is the fundamental difference between these two types of the spores. So intermediate level of these germicides can kill the fungal spores, not the endospores. Now tubercle bacilli and viruses, these are some other organisms which can be killed by intermediate level of germicides. Now they can be used to disinfect the devices that will come in contact with the mucous membrane, but they are not invasive. So this is the way how we can use the different level of concentration of chemical agent for decontamination. Then we'll discuss lastly about the low level of chemical agent. If we are using this type of the low level of chemical agents, then they will eliminate only the vegetative structures that includes the vegetative form of bacteria, vegetative form of fungal cells and some viruses. This can clean the surfaces that touch the skin, but not mucous membrane. Mucous membrane, obviously it is the inner tissue or inner membrane, so that can not be clean, but the upper surface or the skin can be done easily by this type of the low level of the consideration. This desirable qualities for the chemicals. Means what are the qualities what we are looking for when we consider some chemical compound or chemicals that should be used for chemical agent of the antimicrobial things. Then this list of characters or the qualities should be required for this chemical compound said to be antimicrobial agents. So they should be rapid in action in low concentration. It is very important because the action all the time should be very rapid and they should use in very low concentration. Then and then it can be used in a normal condition. Solubility of that chemical compound should be soluble mainly into the water, but if it is not so, then it can be it has to be soluble in alcohol. So we can manage the concentration or a solution of this type of the chemical agent easily because it should be soluble in water or alcohol. Then broad spectrum. What is the meaning of that? Broad spectrum means it should cover range of organisms, that one type of chemical which will be able to kill so many different types of the bacteria or microorganisms. That type of the chemical agent should be taken, or this should be considered as a quality of that chemical. Then low toxicity. The toxicity level should be low, so it can be less harmful for us, those who are using this, and it should be harmful most for the organisms, microorganisms against which we are using them. Then the penetration, that is the penetration power, means it should reach up to the desired level when we are using them on skin or upper surfaces of the non-living material or whatever. So it should be able to penetrate into the layers, and it should be able to penetrate into the microorganisms, and it should be able to kill them. So that penetration power should be higher for the chemical compounds. Non-corrosive and non-staining. That type of the compound should be non-corrosive and non-staining, means they should not give any type of corrosion and not should give any type of the staining. So these are some required qualities for any type of the chemical compound which should be used as chemical agents for controlling the microorganisms. The factors affecting the germicidal activity of the chemicals, that which are the factors which will affect this germicidal action of the chemical, that the nature of the material being treated, that what we are treating. So for what material we are doing this treatment of germicidal action, that should be considered. Degree of contamination, how much that area or environment is been contaminated with some microorganism, that is to be considered. Time of exposure, that for how much period of time we are using the chemical treatment, that is to be considered. And then strength and the chemical action of the germicide, whatever the type of the chemical compound we are using, what should be the strength and what is the chemical action of that compound, that affects the activity. Now we will have the quick look at the different chemical compounds or the agents which are basically we have distributed them into several parts. That is by this particular slide you will come to know that basic, that is functional groups. I think you all are very clear about that chemistry. That is all chemical compounds are having their particular functional groups, and according to them we have distributed all different types of the chemical agents in different areas. The first, that is phenolics, then quaternary ammonium compounds, peroxygens, halogens, aldehydes, biguanidines, heavy metals, sterilizing gases, dyes, acids and alkalis. So these are various groups what we have distributed over all different chemical compounds, and turn by turn, slide by slide we will go through that what this type of the particular group of compounds will act, means what should be their mode of action, how they will kill the organism, and what are their products. Means I would like to show you also some pictures that products are available into the market, and we are frequently using them in our daily routine. That what sir has also told, that we are using so many chemicals in our laboratory and so many chemicals at our home also for surface cleaning, for cleaning of our hands and all. So we are once again I reminding you that we are not discussing about the medicinal aspects of chemical agent, that is a drug, but we are using discussing about the chemical aspect what we are using for surfaces and the external surfaces of the body and all. So first of all we'll look at the phenolic compounds. This phenolic compounds is it is very much I think clear in your mind that what should be the basic phenolic compound structure of this compound. I will show you the structures in next 2 or 3 slides, but we know that it's an aromatic organic compound with attached OH group. Here that is a OH group has to be attached with the aromatic ring. Now how that type of the compounds will disrupt the microorganism and denature the microorganism, that they will denature the protein part of that microorganisms, means all our organisms possesses numerous amount of proteins in the form of enzymes and different certain organelle parts. So if the protein is been detached, then so many metabolic activities and physiological functions will be disturbed, and ultimately the cell will not be able to reproduce and it will die. So this way phenolic compounds will disturb the microbial activity or inhibit or kills the organism. The examples we will take, that is phenol, orthocresol, orthophenylphenol and hexachlorophene. These are some compounds which are used as phenolic compounds as antimicrobial agents. One most commonly used disinfectants is here we are going to consider or use the term disinfectant, but I don't repeat that this terms has been very much made clear by Rakesh sir. So we are using this Lysol, right, that is tuberculosidal. Now once again this sidal term comes, so that was the aspect to cover the different basic terminologies. So tuberculosidal means it will kill tubercle type of the organisms or bacteria. So these are the action of phenolic compounds. Now we'll discuss about what is the good part, means pros and cons, that how it is effective and what are the limitations for using this type of the phenolic compounds. So effectiveness is what, that they are effective in presence of organic matter, remain on the surface for the longer application, long after the application, means they are effective in organic matter. That is very important aspect of phenolic compounds. And other important aspect is that they remain on the surface for long period of time after the application, so they will give good amount of disinfection action. Now other negative part is what, that phenolic compounds are having some negative part also, that why they are not used very rapidly, because they are having disagreeable odor and skin irritation. Means what, the odor is not that much pleasant, so sometimes people are having this disagreement for the use of phenolic compound. And once hexachlorophene can use for so many for number of time as antiseptic agent, but now its use has been limited because it is also found that sometimes it can cause brain damage. So that is the negative part of using this phenolic compounds. Now these are some structures from which you can have a look at this. So in the central structure, that is only OH is been there and the phenolic ring is there. It's a basic structure of phenol, and there are some other examples of the phenolic compounds also. So you can understand that which type of the chemical structure this type of the phenolic compound possesses. For that explanation I have taken these pictures. In the pictures right to this bottom part you are having that Lysol spray. So that type of the spray is now available into the market. You can apply on any surface, and you can sanitize the floor or something like that, your lab area, your lab table, something like that. So these are the applications of phenolic compounds. Now we'll move further and goes to next category. That category gives you idea, that is alcohols. So so many different type of the alcoholic compounds are there, and they can also be used as antimicrobial agent or the antimicrobial chemical agent. First among them we will discuss, that is ethanol, and other is isopropanol. This ethanol and isopropanol can be used at the concentrations between 70 to 95%. That is only the concentration which can be utilized, and they will be able to kill microorganisms smartly. They will kill the organisms once again by denaturing the protein. I have explained you why due to the denaturation killing occurs. And now other aspect is also here, that they also disrupt the membranes. So membranes, now you all are aware about the function of cell membrane. The function of cell membrane is to control the transport of the nutrients and other things or other chemical compounds. If this is been disturbed, then it also affects the cell's feasibility. So this way they will kill the organisms. They kill the vegetative form or cells of bacteria and fungi, but they will not be able to kill spores. Once again here I want to make you clear that spores, they are bacterial spores, not they are fungal spores, right? So they will not be able to kill spores, so their penetration is comparatively lower, right? They used in the disinfecting surface, means as for example we have quoted, that is a thermometer. So you all are aware that we are using thermometer, and we are keeping the thermometer in our mouth for 1 minute or so, and we are measuring the temperature of the body. So all the time the thermometer, what we cannot directly use without doing any disinfection treatment. So for them this ethanol or isopropanol concentrated material, that is very useful for cleaning or disinfecting this type of the surfaces of thermometer. Then one very important and very, you can say, common use of ethanol, you all might have done this, because we are doing sprayed plate technique in cultivation in our laboratory. So ethanol flaming technique, that particular ethanol flaming technique is used by most of all microbiology students. So they will sterilize their glass spreaders with dipping the spreader into the ethanol and then flaming them in the flames of the burner. So this technique is used very widely. So this can be used by this. So this is the example of alcohols and some alcoholic compounds. So it is this technique is been shown. The hand is been washed with shows the disinfection of skin surface. Then a spreader is been flamed. So this type of the use is been done by the use of the alcohol as a antimicrobial agents. Now halogens, we come to next part, that is the halogens and halogenic compounds are important, and they are also doing the same thing, what is the killing of the organisms by chemical agents. So very important group halogen, very important compound of this group is chlorine and chlorine compounds that can be used in the disinfection in municipal water supplies as sodium hypochlorite, calcium hypochlorite or the chlorine gases. So hypochlorites are the tablet forms. Then sodium hypochlorite, that is chlorine bleach, that can be used at the concentration of 10 to 20%, and so on. Halazone tablets are also been used for the campus to disinfect the water for drinking. So this is the use of this is about the use of chlorine and chlorinated compounds. Then iodine. So it is also very common, and you all might have come to across to this type of the thing, that iodine, that is 3 types of the iodine you are using, that is tincture of iodine. So that what is the tincture? Tincture is the iodine solution prepared in alcohol, and then Ki, that is potassium iodide, that is the aqueous solution of potassium iodide can be used, or iodophore group, that is iodine complex with an organic matter organic carrier, that can be used also as a chemical agent. Used these all type of the mixtures of iodine that can be used as antiseptics for clearing the skin or the surfaces, skin surfaces and wounds. So this is the example that how we are using iodine and different compositions of iodine for different uses. Then heavy metal. As we know that heavy metals are having reverse effect on the growth of microorganism. The heavy metal, this category includes mercury, silver, zinc, arsenic, copper ions, and so on. So they will how they will do this antimicrobial action, that is they are forming precipitates with the cell proteins. So once again by the protein denaturation they will cause the death of the microorganisms. At one time frequently used medically as antiseptic, but now after that it has been replaced by the less toxic alternatives. So as for examples, 1% silver nitrate was used as an ophthalmic drops in newborn infants to prevent gonorrhea, but now it has been replaced by erythromycin or some other antibiotics. So priorly they were using silver nitrate solutions for newborn baby, but now it has been replaced by the antibiotics. Then copper sulfate is the other variety which is been used as algicides in the swimming pools. So this is a plate, and there are three different types of the heavy metals are there. As you can understand and see from the figure or the photograph, that zone of inhibition is been seen in all different types of the heavy metals differently. Quaternary ammonium compounds, this is also a new group, the group of compounds which has been used in the, basically they are the used majorly in detergent industry. That quaternary ammonium compounds are the cationic detergents. They are once again denaturing the proteins and disturbing the membranes, and by this way they will kill the organism and doing the antimicrobial action. They will use as disinfectants and skin antiseptics. The acetyl pyridine chloride, you all are aware about this particular agent, because if you are in SYBSE or in TYBSE, this compound particularly has been used in cell wall staining, that is acetyl pyridine chloride. It is a quaternary ammonium compound and is used in the disinfection very well. So these are some examples of quaternary ammonium compounds, and the product which is used for oral, you can say type of the thing for the penetration. It's a quaternary ammonium compound. Aldehydes, this formaldehyde and glutaldehyde are the common examples for this group. They will react chemically with the nucleic acid and protein and inactivating them. So by inactivation of nucleic acid, you can understand that DNA or RNA will be denatured and deactivated, so the organism cannot be survived. So by this way aldehydes can remove the organisms. Peroxygens and hydrogen peroxide, H2O2. So once again they also damages the protein and DNA, and they are doing this type of the damage to the organism, so they are used for chemical agents. The weak solution up to 3% and the strong solution of 25%, it's utilized according to the need. So strong solutions are sporocidal also, and they should be used in a increasing concentration. Then spores can also be eliminated, otherwise only vegetative form can be eliminated. So H2O2, that is skin and wound cleaner, we are using it very normally. Then peracetic acid, it's a quick method for sterilizing the medical equipment. Then ozone, that is O3, it is used in water treatment. Sterilizing gases, that is the ethylene oxide, that is used for sterilizing the heat sensitive material, particularly the plasticwares, right? You all are aware about the glasswares. You have seen different type of the glasswares, that is beakers and flask and pipettes, normal and all. So that type of the plasticwares are also available. They are many times not autoclavable, so physical agent is not useful for them. So in case of this type of the plasticwares, this type of the heat sensitive this type of the materials, ethylene oxide and all, this type of the gases can be utilized. They can be explosive, so used at a concentration of 10 to 20% mixture, or with the either with CO2 or that with the dichlorofluoromethane. They require a special ethylene dioxide type of the sterilization to control the sterilization condition as well as the extensive ventilation after the sterilization because of the toxicity. So once again we all have to consider that it's very having very high amount of toxicity, so it can be used under the strict supervision. Much of the commercial use for ethylene dioxide that is been done priorly for the plastic petri plates and all, but now in this new era it has been replaced by the gamma irradiation method. So gamma irradiation will also do the same type of the things, but it is once again a physical method for the control of microorganisms. Vapor phase hydrogen peroxide, so that is also other example of this gases that has been used recently to decontaminate the biological safety cabinets. So then lastly, that is the beta propionate. It is in the liquid form. It used to sterilize the vaccines and serums. They will decompose after several hours, and so it is not as difficult to eliminate as ethylene dioxide, but doesn't penetrate as well as the ETU and may be carcinogenic. So these are the drawbacks of beta propionolactone. So it can be not used that much frequently. Now these are all what we have discussed about the various chemical compounds, various group of compounds which can be used as antimicrobial agents. Now which type of the antimicrobial agent should be used, or which will be very beneficial for us? So in that case we have to evaluate the effectiveness of each and every type of the chemical agent. This phenol coefficient test will tell you about that thing. So now I will request Dr. Rakesh Bhai Patel sir once again that they will explain you about the evaluation and effectiveness of different type of the chemical agents. Please sir.
Dear students, as you have seen, Rikin Bhai has discussed in detail about various agents and given you the brief idea about the applications of various agents which are being used at present. What are the advantages and disadvantages of them? But now the question arises, which agent can be used, because we have a list with us, but which one is to be used? That depends upon various factors, and one of them is its potency. So to evaluate the potency or the effectiveness, which one is more effective, that can be done by a test which is known as phenol coefficient test. Now this phenol coefficient test, let me clarify that this is basically comparison of a particular chemical in relation to its effectiveness compared with the phenol. Now this is totally in vitro test, and therefore the results which are obtained always does not necessarily be same in field conditions, that is in actual use. However, still phenol coefficient test is considered to be a good test when we are evaluating a particular chemical. Here what is done is various dilutions of phenol as such is made, and the similar type of dilutions of the experimental disinfectant, means the disinfectant in test, that is in question, are done, and they are inoculated with a standard strain of Salmonella typhi and Staphylococcus aureus. They are incubated normally at 37 degrees centigrade, and the next day the results are seen. The results are interpreted as in form of a turbidity, which is indicated by observation, and then after the results are obtained, that is once the inoculation is done into the phenol and the disinfectant, the organisms are next transferred into the broth, and the results are observed in terms of turbidity. Now these results, what is actually happening is, at different time intervals these cultures are being transferred. So The results are interpreted in the terms that the highest dilution that will kill the bacteria after 10 minutes exposure. So which dilution is able to kill the organisms after 10 minutes of exposures but not after 5 minutes? It means in between 5 to 10 minutes. So that is considered as a phenol coefficient test, and it is interpreted as the reciprocal of the maximum effective dilution for the test disinfectant is divided by the reciprocal of the maximum effective dilution for phenol to get the phenol coefficient. For example, let us say the Salmonella typhi, whose maximum effective dilution for phenol is 1 is on 90, 1 is to 90 or 1 upon 90, while that of the effective dilution for disinfectant is 1 upon 450. So the phenol coefficient disinfectant X is for Salmonella typhi is 450 upon, that is 450 upon 90, that is equal to 5. That means it is 5 times more effective than phenol solution. So these are used for comparing the disinfectant which we want to use, but that is in comparison with the phenol. Next one, which is rather more prevalent, is the use dilution test. Here we take certain glass rods or metal rings which are dipped and dried in a suspension of bacteria. It means either on the rings or on the beads or on the glass rods the organisms have adhered. Now these materials, they are then placed into disinfectants for 10 minutes at 20 degrees centigrade, and later they are transferred to the appropriate media for the growth, where their death or survival is an important growth indication. Third one, which is used, third method is used, which is known as a disc diffusion method or Kirby-Weir method. Here the disinfectant is impregnated into the disc and dried, and these discs are then placed on a medium, that is the agar plate, which has been previously spreaded with the test organism. As you can see in the diagram that various discs have been placed and the zone of inhibition indicates the effectiveness of the chemicals. Larger the zone, better or more effective that particular chemical is. So by comparing this kind of, comparing it with the known disinfectant, the unknown disinfectant or the newly discovered disinfectant, and its efficacy can be measured. So these are the three methods by which we can compare the efficiency of disinfectant. Okay, thank you very much. Before I close, I hope this particular subject would have been of great benefit to your syllabus also, because it is in the curriculum also. At the end, on behalf of Rikin Parikh and on my behalf, I would like to acknowledge the Commissioner of Higher Education and the coordinator of Sandhan program for giving us this opportunity to discuss such a topic among the students. We are also thankful to the staff of the BIOSEC studio who has taken pains to bring this program live on the air. Thank you very much.
Thank you, sir.